Hello students, so our next topic will be features of a cloning vector. That means yesterday we have studied about this cloning vector PBR322. Okay, so in continuation of these vectors we are going to study all the features. So first one will be the origin of replications. You can see that this is the portions where the replication will actually starts in this cloning vector. अब यो this origin of replication भनेको चाहिँ it is actually it is a DNA sequence. यो चाहिँ DNA को sequence हो. So from where the replications of the DNA will start, where the DNA will start making its own complementary DNA. Okay, a piece of a DNA linked to ORI. So we know that in this origin of replications the DNA will be linked. Okay, and that piece has to be replicate inside the host cell. That means in the cell in which the desired DNA will be will be start replicating in numbers of cells, then that cell is known as host cell. So this is also controls the copy of the number of the linked DNA. So for getting many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. That means के हुनु पर्यो, होस्ट सेल चाहिँ यस्तो सेल हुनु पर्यो जसमा चाहिँ के हुन सक्छ, लार्जर नम्बर्स अफ DNA चाहिँ लिङ्क्ड हुन सक्छ. सो द्याट आफ्टर लिङ्क्ड भइसकेपछि चाहिँ टार्गेट DNA के हुनु पर्यो, हाई नम्बर्समा चाहिँ रेप्लिकेट हुन सक्ने सच टाइप अब होस्ट चाहिँ हामीले फर्स्टमा चुज गर्नुपर्छ. ओके, so next is number B is selectable marker. You can see that this is the site for the selectable marker. Okay, ampicillin R and this tetracycline R will be considered as the site for the selectable marker. That means this selectable marker भनेको चाहिँ marker gene. Okay, it helps to select transformant and eliminate the non-transformant. That means यो marker gene will help us to select the कुन चाहिँ gene transfer भयो, okay, transformant भयो, उसकोमा transformation आयो, and which one is non-transformant. यो कुरालाई distinguished गर्नुमा चाहिँ this marker gene will be very helpful. अब transformant र non-transformant, that means के को चाहिँ transformation, केमा चाहिँ transformation आउनु पर्छ भन्ने चाहिँ, this transformation is a procedure, एउटा process हो, एउटा technique हो in which a piece of DNA is introduced into the host bacterium. So that means transformant मा चाहिँ के भएको हुँदो रहेछ, piece of DNA introduced भएको हुन्छ in the host bacterium मा. But in the non-transformant मा चाहिँ this piece of DNA, that is the foreign DNA अथवा desirable DNA अथवा target DNA, interest DNA, whichever जे भन्दा पनि the same thing is there. A piece of DNA चाहिँ introduced भएको छैन host bacterium मा भने that will be considered as a non-transformant. So we have to be focused only about the transformant. Okay, and these selectable markers of E. coli, that is Escherichia coli, will include the genes encoding resistance to antibiotics like ampicillin, chlora, amphenicol, tetracycline or kanamycin. So these are the antibiotics. यो antibiotics को लागि चाहिँ resistance होस् भनेर चाहिँ, okay, त्यसको लागि चाहिँ gene हुन्छ which will be encoding resistance to the antibiotics. र जुन चाहिँ E. coli मा भएको selectable markers मा भएको gene छ, त्यो gene ले चाहिँ if that E. coli will be grown in the medium consisting of these antibiotics such as ampicillin, chlora, amphenicol and tetracycline र kanamycin मा हामीले चाहिँ if we grow that E. coli, then that E. coli will will be not died. This will be like easily grown in such a medium. That is all because of those these genes which is resistance to these antibiotics. बुझ्दैछ कुरा? The normal E. coli cells do not carry these resistance genes against any antibiotics. That means our normal E. coli this is the E. coli. Okay, this is the normal E. coli, and there are two E. coli okay, there are two E. coli. First one is the normal E. coli. This one is normal E. coli, and number two is this will be consisting of a certain selective marker gene. Okay, selective marker gene छ. Normal जुन चाहिँ E. coli छ, यो normal E. coli मा के हुँदैन, this will not have any resistance gene. यसमा कुनै पनि resistance gene छैन number 1 को E. coli मा. Okay, number 1 E. coli लाई चाहिँ if we like put in a certain medium जसमा चाहिँ antibiotics छ, okay, त्यो medium मा चाहिँ E. coli will die as soon as this will be put inside this antibiotics containing medium. Thorough in number 2 type of E. coli, that means which will be consisting of the selectable markers, which will consist selectable marker भनेको पनि एउटा set of gene हो जुन चाहिँ gene ले चाहिँ के कोड गर्छ resistance for the antibiotics को लागि. That means antibiotics को against मा fight गर्नु सक्छ, tolerate गर्नु सक्छ, त्यो ability हुन्छ अब यो E. coli मा, र यो gene चाहिँ उसकोमा छ. So after having this selectable marker gene which will be resistant to antibiotics, if this number 2 type of this E. coli will be grown in a medium containing these antibiotics, then after that this E. coli will not die. Okay, this will normally grow in such type of medium. So that is the main purpose of the marker gene. So यो कारणले गर्दाखेरि हामीले चाहिँ के गर्न सकिन्छ, which one is transformant र which one is non transformant. That means transformant मा चाहिँ के हुन्छ, उसकोमा resistance gene हुन्छ, that is selectable marker हुन्छ. Transformant this will not die in the antibiotics, but this non-transformant will be die in the medium of antibiotics. So from that we can easily select the transformant apart from this non-transformant. I think you all are clear. So third one is the cloning site. Okay, so in order to link, okay, alien DNA, that means the alien DNA भनेको foreign DNA or desired DNA, target DNA, जुन DNA चाहिँ हामीलाई को copies बनाउनु पर्ने छ, that DNA will be considered as a alien DNA. No need to get confused. Okay, the vector needs very few recognition sites for these restriction enzymes. Vectors ले चाहिँ के हुन्छ भन्दाखेरि चाहिँ खाली few recognition sites हुनु पर्छ. Okay, so के हुनु भएन, थुप्रो recognition sites हरु चाहिँ हुनु भएन, only single recognition sites हुनु पर्छ for this restriction enzymes. अब किन चाहिँ single recognition sites चाहिँ हुनु पर्छ भन्दा, because if there is the presence of the more than one recognition sites, then this will ultimately, or this will generate several fragments which complicates the gene cloning. जति बेसी fragments हुन्छ त्यति बेसी complications हुन्छ gene cloning को लागि. यो complications नआओस् भनेर चाहिँ only single recognition sites मात्रै हुनु पर्छ for restriction enzymes, जहाँनिर चाहिँ restriction enzyme ले आएर चाहिँ त्यो segment लाई cut गर्छ र त्यहाँनिर desirable alien DNA will be inserted. Okay, and the ligations of alien DNA, that means the joining of, that is the adjoining of this alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. So we already know that ampicillin R and TET R are the this AMP R is the resistance for ampicillin antibiotic, and TET R is resistance for the tetracycline okay antibiotic. So you दुईटा antibiotic resistance genes हरु present हुन्छ PBR322 मा, तर यस मध्येमा चाहिँ एउटा रेसिस्टेन्स जिनमा चाहिँ मेबी एम्प रेसिस्टेन्समा पनि हुनसक्छ टेट आरमा पनि हुनसक्छ दुईटामा चाहिँ एउटा जुन चाहिँ यो जिन छ एन्टिबायोटिक रेसिस्टेन्स जिनमा चाहिँ दिस लाइगेसन अफ द एलियन डिएनए okay that means we have two from the beginning of the class we have been discussing about two antibiotic resistance genes छ okay tet r र emp r छ यो दुईटामा चाहिँ एउटामा के हुन्छ अब एलियन डिएनए चाहिँ हामीले के गर्नुपर्छ एड गर्नुपर्छ okay for this adding of the alien DNA for example ligation of that means ligation भनेको चाहिँ addition join हुनु ligation of alien or foreign DNA is the same thing at the BAMH1 site of tetracycline resistant genes in the vector PBR322 if we talk about the vector PBR322 मा हेर्नु पर्यो भने चाहिँ अब हाम्रो alien DNA अथवा foreign DNA चाहिँ कहाँनिर ligate हुन्छ भने this foreign DNA will be ligated in the site called BAMH1 मा of the tetracycline resistance gene मा चाहिँ हाम्रो insert अथवा ligations हुन्छ once this foreign DNA will be inserted into the tetracycline resistance genes त्यतिखेर चाहिँ now this recombinant plasmid को एउटा जुन दुईटा एउटा एबिलिटी थियो tetracycline र ampicillin मा रेसिस्टेन्स गर्ने र यहाँनिर अब के हुन्छ भने now this DNA okay this recombinant plasmid जसमा चाहिँ अब foreign DNA चाहिँ tetracycline resistance साइट मा आएर जोइन भएको छ okay they will like lose tetracycline resistance अब tetracycline को medium मा grow गर्यो भने चाहिँ that will not live in the tetracycline resistance medium मा चाहिँ okay but they can be selected out from non recombinant र पनि अझै पनि हामीले non recombinant बाट उलाई select गर्न सकिन्छ अरे okay once by plating them in plating the transformant on ampicillin containing medium okay it's very simple कि simple मा बुझ्नु पर्दाखेरि चाहिँ like यो प्लेस मा चाहिँ के गर्दै थियौ अघि देखि चाहिँ we are like के गर्यो भन्दाखेरि चाहिँ हामीले okay BAMH1 मा we have inserted this foreign DNA अब जब foreign DNA BAMH1 चाहिँ कुन चाहिँ साइटमा छ tet r that means tetracycline resistance को एउटा सिक्वेन्स मा चाहिँ हामीले जब इन्सर्ट गर्छौं त्यतिखेर चाहिँ अब यो PBR322 चाहिँ will be no longer resistance to the tetracycline जुन चाहिँ एन्टिबायोटिक मा अब रेसिस्टेन्स हुँदैन उसको एउटा एबिलिटी लस्ट भइसकेको छ र पनि हामीले के गर्न सकिन्छ we can easily select this recombinant out from this non transformant कसरी भन्दाखेरि चाहिँ after okay plating this transformant on ampicillin containing medium किन भन्दाखेरि चाहिँ हाम्रोमा एउटा मात्र रेसिस्टेन्स सिक्वेन्स थिएन we have two resistance sequence that is ampicillin resistance छ सो यदि हामीले चाहिँ यो जुन चाहिँ recombinant DNA लाई चाहिँ if we plate on the medium containing ampicillin resistance त्यतिखेर चाहिँ हामीले यसलाई छुट्याउन सकिन्छ किनभने यसकोमा अझै एउटा एबिलिटी छ that is resistance towards another type of antibiotic that is ampicillin resistance okay after ligations of this all thing अब त्यसको बादमा चाहिँ we have to transform this transformant into the tetracycline medium मा अब tetracycline medium मा लगाइसकेपछि चाहिँ the recombinant grow in ampicillin medium but not in the tetracycline medium अब जुन चाहिँ ampicillin medium मा grow गर्दै गरेको जुन चाहिँ के थियो recombinant थियो recombinant means having the sequence of DNA from the two different types of DNA अथवा source बाट जुन चाहिँ DNA combine गरेर बनिएको छ that DNA is known as as the recombinant DNA अथवा transformants पनि भन्नु सकिन्छ तर यो केमा बाँचेन टेट्रासाइक्लिक मिडियममा चाहिँ सर्भाइभ गरेन but non-recombinant will grow on the both medium containing both the antibiotics किन भन्दाखेरि यो non-recombinant मा चाहिँ के छ this non-recombinant will be having both type of ampicillin and tetracycline resistance तर recombinant मा चाहिँ जब के आउँछ another foreign DNA आउँछ त्यतिखेर यो recombinant DNA बन्यो recombinant DNA will be only survive in the ampicillin medium but will not survive in the tetracycline medium only the non-recombinant जसमा चाहिँ कुनै पनि recombination छैन so यो non-recombinant मा चाहिँ both the medium मा these cloning vector will survive So in this case, अब यो केसमा चाहिँ हाम्रो के हुँदैछ भने antibiotic resistance genes help to select transformant, right? Whereas the other antibiotic resistant genes get inactivated due to insertions of alien DNA and helps in selections of the recombinant DNA. That means, so out of these two antibiotic resistant genes, यो antibiotic resistance gene मा चाहिँ एउटा inactivated हुँदैछ due to insertions of DNA. Okay, alien DNA तर अर्को चाहिँ एक्टिभेट भएको कारणले गर्दाखेरि चाहिँ we can easily select which one is a transformant हो भनेर select गर्न सकिन्छ okay so one resistant gene चाहिँ inactivate हुन्छ so selections of recombinant due to inactivations of antibiotic requires simultaneous plating of two plates having different antibiotics that means एउटा दुईटा यस्तो प्लेटिङ हुनु पर्यो यो दुईटा प्लेटिङमा चाहिँ दुईटै एन्टिबायोटिक हुनु पर्यो एम्पिसिलिन पनि हुनु पर्यो टेट्रासाइक्लिन पनि हुनु पर्यो किनभने चाहिँ एउटा रेसिस्टेन्स जिन चाहिँ हाम्रो के भएको हुन्छ इनएक्टिभेट भएको हुने चान्सेस हुन्छ अब जुनै पनि हुन सक्छ त्यो कारणले गर्दाखेरि चाहिँ हामीलाई दुईटा प्लेटिङ चाहिन्छ Therefore, alternative selectable marker जुन चाहिँ छ alternative selectable markers have developed to differentiate recombinant from non-recombinant. So यो जुन चाहिँ हाम्रो अगाडिको प्रब्लम्स भएको कारणले गर्दा so later on के डेभलप्ड भएछ alternative selectable marker डेभलप्ड भयो. So यो alternative selectable marker ले चाहिँ के हेल्प गर्छ भने which will be developed to differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate. So यसमा चाहिँ we don't have to plate any antibiotic. यसमा चाहिँ we just only need certain enzymes, okay, certain substrates मात्रै चाहिन्छ so that enzymes ले त्यहाँ काम गर्छ. यदि color present भयो अथवा color absence so in the presence of the color the absence of the color ले मात्रै अब चाहिँ which one is a recombinant and which one is non-recombinant भन्ने we can easily distinguished in the alternative selectable marker मा चाहिँ okay or recombinant DNA अब जुन चाहिँ DNA recombinant भएको छ दुईटा DNA मिलेर के बनिएको छ भने एउटा ओके 2 DNA बाट चाहिँ एउटा न्यू DNA बनिएको छ that DNA is the recombinant DNA अब उस is inserted within the coding sequence of enzyme एउटा enzyme that is a galactosidase is the enzyme यो enzyme को एउटा coding sequence that means the एकदमै जसबाट चाहिँ केही कुरा सिन्थेसिस हुन्छ ओके केही कुरा बनिन्छ that is the coding sequence a functional sequence is known as a coding sequence यसमा चाहिँ this recombinant gene will be inserted after that the enzyme is inactivated okay it is called insertional inactivations so such colonies do not produce any color these are identified as recombinant colonies that means जुन चाहिँ enzyme called ए ग्यालेक्टोसाइडेज एन्जाइमको कोडिङ सिक्वेन्स फङ्सनल सिक्वेन्समा चाहिँ वी हेभ इन लाइक इन्सर्टिङ रिकम्बिनेन्ट डिएनए। जब यो रिकम्बिनेन्ट डिएनए इन्सर्ट गरिसकेपछि चाहिँ एन्जाइम ए ग्यालेक्टोसाइडेज विल बिकम इनएक्टिभेटेड। द्याट मिन्स इन्सर्सनल इनएक्टिभेसन भन्नु सकिन्छ, जसमा चाहिँ रिकम्बिनेन्ट डिएनए इन्सर्ट गर्दा चाहिँ इनएक्टिभेसन भयो। सो सच टाइप्स अफ कोलोनिज विल नट प्रड्युस एनी कलर। यो कारणले गर्दाखेरि चाहिँ यदि कलर प्रड्युस्ड भएन भने चाहिँ दिस कोलोनी विल बी द रिकम्बिनेन्ट टाइप अफ कोलोनी भनेर वी क्यान इजिली प्रेडिक्ट। ओके, इफ द प्लाज्मिड इन ब्याक्टेरिया डु नट ह्याभ एनी इन्सर्ट, यदि हामीले कुनै पनि रिकम्बिनेन्ट डिएनए इन्सर्ट गरेको छैनौँ भने चाहिँ द्याट टाइप अफ प्लाज्मिड विल बी giving blue colored colonies in the presence of a chromogenic substrate. Okay, this chromogenic means color creating substrate, and ani hamile chai okay this one is non recombinant bhanera we can easily understand.